Transposons in prokaryotes and eukaryotes. Transposons in prokaryotes. There are two types of transposons in prokaryotes. They are called as insertion sequences or IS and transposons like TN and bacteriophage MU. So in case of prokaryotes, there are two types of the transposons, insertion sequences and transposons like TN or MU. Transposons in prokaryotes, what are IS elements? IS elements, prokaryotic IS elements range in size from 768 base pair up to 5 kb. Like the example is of IS1 is 768 base pair long and present in 4 to 19 copies of E. coli chromosome. Another example of IS sequence is IS2 that has 0 to 12 copies. Another example IS10 which is found in R plasmids. What is IS element and how it looks? The ends of all sequences mean IS elements show inverted terminal repeats of 9 to 41 base pair. Like the example is IS1 has 23 base pair of inverted repeats. This is the diagram of insertion sequence or IS sequence where there is a transposase gene and both sides there are inverted repeats. So these are the inverted repeats on this is GG, TGA, so on, and same is here GG, TAA, and so on. So on both ends in case of IS elements there is a transposase gene and inverted repeats. Transposon in prokaryotes IS elements. In case of IS elements, there is a gene in the middle and on both ends there are inverted repeat sequences. Transposons in prokaryotes. Transposons are similar to IS elements but carry additional genes and have a more complex structure. There are two types of prokaryotic transposons. First type is called as composite transposons and second type is called as non-composite transposons. In case of composite transposons, they carry genes like antibiotic resistance genes and flanked on both sides by IS elements. While in case of non-composite transposons, they carry genes like drug resistance but they do not terminate with IS elements. So this is the difference between composite transposons and non-composite transposons. In case of composite transposons, they end with IS elements, while in case of non-composite, they do not end with IS elements. Transposon in prokaryote, the example of composite TN10 element. Here we can see that this is the transposon TN and here is a gene for tetracycline resistance and on both ends there are IS elements. Example of non-composite transposons. The example of non-composite transposon is transposon TN3. Here we can see that there are additional genes lactamase, then resolvase and transposase. And on both sides there are no IS elements. There are inverted repeats but there is no IS elements. So this is the difference between composite and non-composite transposons in prokaryotes. This is another example of non-composite transposons and the sequence of portion of left inverted repeat and the portion of right inverted repeat which is present on both ends of the transposon. Transposon in case of eukaryotes. In case of eukaryotes, the example is of TY elements in yeast. So this is the diagram of yeast transposon elements that is TY element. In the middle, there is a transposase encodes two portion that is RNA and on both sides there are long terminal repeats. Long terminal repeats on both sides, that is approximately 5,900 base pair. So this is a typical Ty element which is found in the yeast. In case of eukaryotes or higher eukaryotes, there are activator and dissociation elements. So activator, this is the diagram of the activator elements, and these are the dissociation elements. These type of the transposons, they are found in higher eukaryotes. Like the example can be of the maize. So transposons in eukaryotes, activator and dissociation elements. The detail of what are activator and what are dissociation elements. Activator elements can be up to 5,563 base pair with 11 base pair imperfect terminal IR sequences. While in case of AC activates DS to transpose or break the chromosome where it is inserted, while the DS elements vary in length and sequence, and all have the same terminal IR and AC, and many are deleted or rearranged versions of activators. So if we conclude that what are transposons? Transposons, they are found in both in case of prokaryotes and in case of eukaryotes.